Okay, folks, so this is 28-2, and it's going to take a little bit of time because I have to teach you meiosis. And when we do and I'm teaching you meiosis for spermatogenesis in this part. When we do oogenesis, you will have already learned meiosis for spermatogenesis, and you'll just learn some of the specifics of oogenesis. So 28-2 will take a little longer than 28-3, where we talk about oogenesis. All right. So first of all, in the fetal development, I'll show you how the testes develop. Uh, the default route of development is as a female. All of us start out developing as a female. And then those of us who have a Y chromosome and the SRY gene, that's what the Y chromosome has. It's called the SRY gene. We release a couple hormones, a couple factors that lead to male development. So we all start out as females, and then those with a Y chromosome and the SRY gene secrete a factor that leads to the male development. Now here's the testy right here. It's up next to the kidney. Initially, it's up next to the kidney. And what happens is there's a connective tissue holding it in place. And this is connected to the bottom of the peritoneum. All right. This connective tissue doesn't contract until about the 7th month or so. But what happens is it doesn't grow either, and the baby grows, and this connective tissue stays the same length. So the testes are pulled downward relative to the baby because the baby is growing taller and taller. And that's what you see here. You see that they're showing you 5 millimeters right here, and then they're showing you 5 millimeters right here. This testy hasn't really moved, but it's moved relative to the baby's body. It's connected to this connective tissue called the gubernaculum testes. By the way, that's it right here, gubernaculum testes. But then around 7 months, the gubernaculum testes does contract, and it pulls the testes down into the scrotum. Because on average, the testes have to be about 1.1 degrees centigrade less than body temperature. So if normal body temperature is 37.2, then your testes want to be about 36.1. All right, about 36.1 degrees Celsius. That's the gubernaculum testes, making sure that your testes do descend into the scrotum. When your testes don't descend into the scrotum, it's called cryptorchidism. Cryptorchidism is undescended testes. Oh, I forgot a P there. Sorry, that's spelled wrong. Cryptorchidism. Cryptorchidism. And the doctors won't do surgery immediately. They'll give the testes time to descend. But usually before puberty, if they haven't descended, they'll go in and surgically make them descend. I do know of a couple cases where the one testicle descended, one didn't. The one that didn't became abnormal and had to be removed. So it actually was precancerous even. So they removed that before puberty of the child. I know in another case, it was a brother of this individual. Um, the brother also had one testy undescended, and but they went in and surgically did descend it, and it wasn't abnormal, so they were able to get it into the scrotum. So this shows you just another picture of the gubernaculum testes. This shows you where they start up near the kidney, so you can see the kidneys here, and then you can see the testes here. And then you can see the testes here and the gubernaculum testes keeping them in place. Baby's growing, they're not moving. That's 4 months. And then at 7 months or so, the gubernaculum testes does contract and pull it down. And here you have it in the scrotum. Okay, so that's that's descent of the testes in cryptorchidism when it fails. There's the collage for you showing the whole thing. All right, so the testes is where the spermatogenesis is going to occur. So we're going to be looking at this area in detail. Before I get there, we studied some of this already. In the dermis of the scrotal skin right here, there's a dartos muscle. This causes the wrinkliness of the scrotum, It's actually within the dermis of the skin. And then deeper to that is this cremaster muscle. Now both of these muscles are involved in pulling the testes up towards the body for temperature regulation or during ejaculation. The cremaster muscle contracts during ejaculation. So these have a couple different functions, but the main thing is, your testes want to be 1.1 degrees, negative 1.1 degrees, so 1.1 degrees less than body temperature, and body temperature is 37.2. So these muscles pull the testes up to your body if they're too cold, and then they relax and let them hang further from your body if they're too warm. So there's those muscles. And then we start getting to the details of the testes, because this is where spermatogenesis is going to occur. So first of all, I think I told you that there's these are seminiferous tubules, and that's where spermatogenesis occurs. Each seminiferous tubule is about 3 feet long. So this one's about 3 feet long, 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 etc., etc. Total, we have about half a mile of seminiferous tubules, which I think, well, I forgot the math. If a mile is 5,280 feet, then half of that is about what? 2,500 is half, 140, 640, 2,500, 2,640. I hope I did that math correctly before that. 2,640 feet of seminiferous tubules. So we have 2,640 feet of seminiferous tubules. But each tubule is only about 3 feet long. So we have, how many of these different tubules do we have separated by septate? There's septum in between each of these. These are called lobules, by the way. So how many lobules do we have? Well, we have about 300 lobules in one testy. So we have about 900 lobules total. I'm sorry, 600 lobules total, because we have 300 or so per testy. That's 602 testicles times 3 feet. Well, that doesn't quite work out, does it? That's only 1,800 feet. So I'm short. Well, the math is not working out. So I have, these are all approximates, by the way. So when I'm saying I have about 3 feet, it could be 4 feet. When I say I have about 300 lobules, it could be 400 lobules. So I have 300 or so lobules per testy. The length of one seminiferous tubule is about 3 feet. In total, I have about a half a mile of seminiferous tubules. I guess trying to prove it to you with the math is because I'm doing too much estimation here. These are all estimates. All right, so that's the testes. We're going to do a cross-section of these seminiferous tubules and look at where spermatogenesis occurs. This is a cross-section. Well, this is just a cut, but because they're so coiled, the seminiferous tubules are so coiled, we get many, many cross-sectional views of it because they coil so much. We get some longitudinal sections in here, but most of the sections are cross-section. This is showing you another cross-section blown up. These are going to be sperm. They're not sperm yet. They're maturing. But this is what's going to be sperm right here, all through here. Okay, so this is the artistic drawing of it, and this is going to be easier to study on here. So this is a seminiferous tubule. This is the lumen of the seminiferous tubule. There will be some fluid secreted into the lumen by the seminiferous tubule itself. We have a few different types of cells here. First of all, we have this nurse cell, very important cell. The nurse cell has a couple other names. The nurse cell is also called the sustentacular cell. Sustentacular. It's also known as the Sertoli cell. The nurse cell secretes antigen binding protein. It secretes inhibin. We'll talk about inhibin in a second. So antigen binding protein and inhibin, it secretes those. Androgen binding protein, I should spell that out for you. Binds testosterone. And these nurse cells support spermatogenesis. So these nurse cells are very important, and these germ cells dividing and going through what I'm going to explain to you in a second, mitosis and meiosis, and eventually becoming a sperm. So the nurse cells are extremely important here. Then we have interstitial cells. Well, the interstitial cells are between seminiferous tubules. So this would be an interstitial cell, and these would be interstitial cells, and these would be interstitial cells, and these would be interstitial cells. Interstitial cells make testosterone. Testosterone. And they're in between seminiferous tubules. Let me back off the view here. Back off the view some more. So you can see that this is one seminiferous tubule right here. And here's another seminiferous tubule right here. So we have these interstitial cells in between. You can see those interstitial cells in between. They make testosterone. Okay. Even a bigger blown up picture, but it allows us to see the blood testes barrier. So you can see the interstitial cells here. They're going to make testosterone. You can see the nurse cell here. And look at this dotted line. What it's telling you is that there's a blood testes barrier. And that means there's tight junctions in here between nurse cells. So between this nurse cell and this nurse cell, there's tight junctions. So nothing can get through down to these developing sperm unless it goes through a nurse cell. So the nurse cell gets a chance to detoxify or eliminate any kind of chemical that could assault the sperm. So there's a blood testes barrier. These cells up here are called germ cells, or also known as spermatogonium. We keep a population of spermatogonium here all the time. And when you're going to make sperm, the spermatogonium goes through mitosis. So now you have two of them. And then one of them, like say for example right here, this used to be one spermatogonium. It went through mitosis and became two. And then one of them stays here to be a spermatogonium, to remain a germ cell, a spermatogonium. And the other one crosses the blood-brain barrier and proceeds through meiosis and becomes four sperm. But this one that stayed up here can go through mitosis again and become two again. So we always keep a population of spermatogonium in our seminiferous tubules up here. Okay, so we're going to go through the details of spermatogenesis here shortly. All right, so we have to have, here's what spermatogenesis is. It's divided into this. We have mitosis. The spermatogonium have to go through mitosis. Then we have meiosis. One of the daughter cells of the spermatogonium goes through meiosis. And then we have spermiogenesis. Now, spermiogenesis is one part of spermatogenesis. All three of these things are spermatogenesis. What spermiogenesis is the spermatids maturing. That's what that is. So don't confuse spermatogenesis with spermiogenesis. Spermiogenesis is the spermatids maturing. Okay, so the three steps of spermatogenesis, we're going to look at each step separately. Mitosis, meiosis, and spermiogenesis. Okay, let's do mitosis first. Here's your spermatogonium. And what's going to happen is it's going to go through mitosis, and you're going to have two identical daughter cells. One of these cells is going to go through meiosis. The other stays in the on the blood side of the blood testes barrier as a spermatogonium. If you look at what happens to chromosomes during mitosis, first of all, they double, and then they get pulled apart into separate cells. Now, let's do some nomenclature here. This black one and this gray one are called homologous chromosomes. One came from your mom and one came from your dad. This red one and this orange one are called homologous chromosomes. One came from your mom and one came from your dad. This dark blue one and this light blue one are called homologous chromosomes. One came from your mom and one came from your dad. Homologous chromosomes are not identical. Now, let me be clear here. They have homologous genes or alleles that code for the same characteristic. But for example, if they both code for height, let's just talk about the characteristic height. One could say tall and one could say short. Let's look at another homologous allele. If they code for fur color, one could say brown and one could say white. So homologous chromosomes aren't identical. They are not identical. But they do code for the same traits. Now, when I double my DNA, when I do DNA replication and double it, these two are called sisters, whereas these two are called homologs, like we said before. It's crucial that you know the difference between sister chromatids and homologous chromosomes. Let me say that again. It's crucial you know the difference between sister chromatids and homologous chromosomes to understand mitosis and meiosis. In mitosis, we just double our DNA. We have sister chromatids now. We pull apart sister chromatids to different cells. So these two sisters are going to be pulled apart. One went here, and the other went here. They're in different cells. These are going to be in different cells. So in mitosis, we pull apart sister chromatids. Sister chromatids are identical because when you copy your DNA, if you had a big T here, well, now you have a big T here. And if you have a big B here, now you have a big B here. Sister chromatids are identical. Homologous chromosomes are not identical. All right, now let's talk about meiosis. First of all, you start with a diploid cell. Oh, by the way, let's talk about that word. What the heck does it mean to be called diploid? Diploid means you have homologous pairs. That's what diploid means. You have homologous pairs. So most of our cells are diploid. We have one number 1 from our dad and one number 1 from our mom. We have one chromosome number 2 from our dad and one chromosome number 2 from our mom. One number 3 from our mom, one number 3 from our dad, all the way up through. So diploid means you have homologous pairs. Haploid means one chromosome. One chromosome, no pairs. Doesn't mean one. I shouldn't say it that way. For example, our haploid cells have 23 chromosomes, but no pairs. Haploid means no homologs. A single copy of each chromosome. Single copy of each chromosome. Okay, that's what haploid means. No pairs. All right. So you guys can get that written down. So that's the difference between haploid and diploid. We write haploid N and we write diploid 2N. So you can see that this cell right here is diploid or 2N. What's the first thing we do? The first thing we do is now we're doing meiosis. We start with a diploid cell. The first thing we do is we copy our DNA. So these two are homologs. Those are homologs. We copy them. These two are sisters. And the two gray ones are sisters. But the black ones to the gray ones are homologs. So we copy our DNA. All right. And here we have a cell with the DNA copied. Now, this happens to be a very special phase right here where you make tetrads. And it's called prophase 1. And it's very special. Well, first of all, let me back up. You do meiosis 1 and you do meiosis 2. Prophase 1 is a phase within meiosis 1, and that's why we call it prophase 1. This is a very, very special phase, prophase 1, because we make these tetrads. And something very special can occur in prophase 1, and that is crossing over can occur. And crossing over is when these homologous chromatids, so a piece of the gray can swap with a piece of the black, and a piece of the red can swap with a piece of the orange, and a piece of the dark blue can swap with a piece of the light blue. These homologous pieces can swap. And now you're probably going to see this already. At this point in time, you're going to say, well, if crossing over occurs, then my sister chromatids are no longer identical. And I would agree with you. It's only during meiosis, prophase 1 and meiosis 1 where crossing over occurs. And then at that point in time, sister chromatids are no longer identical. Okay, so that's prophase 1. Then what we do is we finish dividing our chromosomes into 2 separate cells. Now, what do we pull apart? And the answer is, during meiosis 1, we separate homologs. See, that's different than mitosis, because in mitosis, we separated sisters. But during meiosis 1, we separate homologs. So here's a big question. Is this cell haploid or diploid? And is this cell haploid or diploid? And the answer is it's haploid. You're going to say, no, I see 2 blacks and 2 reds and 2 blues. Yeah, you do. You see sister chromatids in each cell, but you don't see any homologous pairs. There's no homologs. And the definition of diploid was having homologous pairs. Where's the homologous pair to that one? It's in this cell. It's in a different cell. So this cell is haploid. It has a problem still, and its problem is that it has sister chromatids that have to be pulled apart, but that's a haploid cell. Because of that, meiosis 1 is called the reduction division. It's called the reduction division. Meiosis 1 is the reduction division. Then, after we have these 2 cells following meiosis 1, they simply go through meiosis 2 and pull apart sisters. Meiosis 2 is a lot like mitosis in that way. They pull apart sisters into 2 separate cells, and now you have your 4 haploid cells. And this is called the equational division. You're just pulling apart sister chromatids. We call this the equational division. But the reduction division is meiosis 1. All right, so that's spermatogenesis. That's meiosis of spermatogenesis. I want to come back and talk about prophase 1 in a little bit and crossing over. So we'll come back and talk about that. This compares mitosis and meiosis for you. So this is showing you mitosis right here, and this is showing you meiosis right here. Let me reiterate the big things here. First of all, prophase 1 and crossing over only occurs in meiosis. I mean, prophase occurs in mitosis, but not crossing over. Prophase 1 is a very special phase in that synapsis occurs. Synapsis is a verb meaning you make a tetrad. So synapsis is a verb saying a tetrad forms. And then crossing over is a verb meaning you can swap pieces of these homologs here. Meiosis 1 is the reduction division. Meiosis 2 is the equational division. This is another picture showing you that. Just another way of looking at it. By no means is it... well, actually, it is a little better, I think. I think it's better than the previous picture. But here's comparing mitosis and meiosis, another picture comparing it. All right. Now, let's go step by step. This is mitosis. Remember, there's 3 steps to spermatogenesis. There's mitosis, meiosis, and spermiogenesis. Now we're going to do the details of it. So this is mitosis. Here's my spermatogonium. It divides. One of the spermatogoniums stays where it is. The other one's about to go through meiosis. We call this the primary spermatocyte. Right here, primary spermatocyte. It is a diploid cell. So my primary spermatocyte is a diploid cell. That primary spermatocyte now crosses the blood-brain barrier. All right. Here's my primary spermatocyte. It's continuing to go through meiosis. It doubles the DNA. and I get my tetrads during prophase 1. This is where crossing over could occur. All right. Then I finish meiosis 1. This is the end of meiosis 1 where I pull apart homologs. So I separate homologs right here. Now remember, a primary spermatocyte was diploid. Now I have my secondary spermatocytes. They are haploid. They still have sisters, but they're haploid. My secondary spermatocytes are haploid. This is the reduction division right here. So from primary spermatocyte to secondary spermatocyte, I have a reduction division. Then I do meiosis 2 and I pull apart my sisters, and these are my spermatids. These are not mature sperm. These are immature sperm. They're called spermatids. Matter of fact, they're still connected. They're connected by gap junctions at this point right now. But those junctions will disintegrate, and these will become individual cells. All right. Now look at this. Well, actually, let's finish this, and then we'll go back and look at it again. And then I have spermiogenesis, and this is the physical maturation of the spermatid. You have to make a nice, well-developed midpiece that has all your mitochondria. Your midpiece has all the mitochondria. You have an acrosome, a well-developed acrosome right here, which is the enzymes. I can't spell. The enzymes necessary to digest their way into the egg, because these are going to fertilize an egg. So you have a well-developed acrosome right there. And of course you have a well-developed flagella, which is going to help it swim. So that's spermiogenesis, or the physical maturation of it. All right. Now I want to go back and show you something. To make your primary spermatocyte, it takes about 2 weeks. Let's keep track of this, all right? We can do the days. It says days right there. So to make your primary spermatocyte from spermatogonium to primary is 16 days. So let's write that right down here. From germ cell to primary is 16 days. All right. From primary to secondary is 24 days. So now you're up to 40 days. So from germ cell to secondary, you've taken more than a month. You're at 40 days now. All right. So you have your secondary spermatocytes now. You've taken 40 days to do this. And then to finish meiosis, it only takes a few hours. So now you're 40.3 days. It only takes a few hours to do that. So now I have my spermatids. All right. And then how long does spermiogenesis take? That takes almost another month. So now it takes 24 days. So now I'm at 64.3 days. So the entire thing takes about 64.3 days. All right, so that's to make a mature sperm, a sperm that's able to swim. All right, let's go back and look at that and see if there's any. Make sure you know this. I'm not doing this for my own benefit. I need you to know this. So from spermatogonium to primary spermatocyte, it takes a couple weeks. So just say, if you want to do it in days, say 16 days. If you want to do it in weeks, say a couple weeks. From primary spermatocyte to secondary spermatocyte, it takes about 4 weeks or 24 days, or approximately. When I say weeks, I'm approximating it. So now I'm at about 6 weeks or 40 days. 7 times 6 is 42, 40. So you can say 6 weeks if you're doing it by weeks, or you can say 40 days approximately. This is all approximations. All right. And then from our secondary spermatocytes to our spermatids, this is only a few hours. Now you're just pulling apart sisters. So right back here, I say 40 days or 6 weeks. I'm 6 weeks in a couple hours. Or you could say 40 point. You can actually say 41 days if you wanted. You could round this to 41 because this is approximation. We can make it easy. You could say 41 days. All right. So you could say 6 weeks and 1 day, or you could say 41 days, something like that. You're still in the ballpark. And then to do spermiogenesis, to mature your sperm into swimmers, it takes another 24 days. So this is, and by the way, your mature sperm is called a spermatozoa. So you're talking 64 days here total. All right, that's the timeline. A little over 2 months to make a spermatozoa. This is the collage. Look at the time. Make sure you know that. Make sure you know this. Primary spermatocyte is diploid. Secondary spermatocyte is haploid. Meiosis 1 is the reduction division. Meiosis 2 is the equational division. Spermatids have to go through spermiogenesis to become spermatozoa. Make sure you know this. This is a slide you have to know all the details on. This is another way of looking at it. Why I brought this picture in is because it shows you the maturation of spermatids to spermatozoa. You can see they have to grow flagella. You can see they have to form their nice mature midpiece with packed full of mitochondria. The N in here means haploid. That's what that's telling you. And then you have your spermatozoa that's capable of swimming. Okay. That is spermatogenesis in a nutshell. Okay, nurse cells, also called sustentacular cells, also called Sertoli cells. This is what nurse cells do. They help maintain that blood-testis barrier. We already talked about that. Very important job of the nurse cells. They support this mitosis and meiosis, and they support spermiogenesis. In other words, they support spermatogenesis, because 2 and 3 here is spermatogenesis. They make androgen binding protein, which binds testosterone, and they make inhibin testosterone. This ultimately, inhibin goes up to the pituitary and reduces FSH and LH. So the more sperm you make, the more you turn off the signal to make sperm. FSH tells you to make sperm, LH tells you to make testosterone. The nurse cells make androgen binding protein, which binds testosterone. So the testosterone that the interstitial cells make binds the ABP of the nurse cells and tell you to make sperm. But it's self-regulating, because when you are activating the nurse cells, they make inhibin, which shuts down FSH and LH. So you can see that there's a negative feedback loop here that shuts down sperm production. So, I mean, what's the whole deal with that? Well, we have about 20 to 100 million sperm per milliliter. And we have about 3 to 5 mils of ejaculate. So in one ejaculation, you can do the low end. 3 times 20 is 60 million. In one ejaculation, you have 60 million to do the top end. 5 times 100 is half a billion. So 60 million to half a billion. 500 million is half a billion. In one ejaculate, you have 60 million to 500 million sperm. Now that's 20 to 100 million per milliliter, and you have 3 to 5 milliliters per ejaculate. All right, so you could have half a billion sperm in your ejaculate. Now, why is this number always about the same? Well, FSH tells your testes to make sperm. Follicle stimulating hormone says to your says to your nurse cells and your germ cells, spermatogonium, make sperm. Luteinizing hormone, also known as interstitial cell stimulating hormone, tells your interstitial cells between your seminiferous tubules to make testosterone. These make the testosterone, which also enable the nurse cells to support spermatogenesis, and you meanwhile make inhibin. Your nurse cell makes inhibin, which goes up to the pituitary and shuts this whole thing down. That keeps your sperm count around this number right here. All right, this Mullerian inhibiting factor. This is what makes you male. We all start off as females. And I have a picture. I'm going to talk to you more about this Mullerian inhibiting factor. The Mullerian, there's these ducts. As a fetus, you have these ducts called mesonephric ducts. And they can form into fallopian tubes if you become a female. And unless you have this Mullerian inhibiting factor, and then the ducts form into vas deferens, not fallopian tubes. So this is what makes you male, and make sure that you don't become female if you're XY. Okay. This is a picture showing you the negative feedback from inhibin. Inhibin comes up in the dotted line, by the way, there's inhibits. You can see that there. Inhibin comes up and shuts down FSH and LH. It's showing you what FSH and LH do. FSH goes down and says to the nurse cells, support spermatogenesis. LH comes down to the interstitial cells and says, make testosterone. What does testosterone do? Well, it gives you your secondary sex characteristics, but it also goes down and tells you and turns on spermatogenesis. But the more spermatogenesis you turn on, the more inhibin you make. So it is a self-regulating cycle here. This is showing you the same thing, but it's talking about the androgen binding protein. The testosterone is carried by this this binding globulin in the blood. All right. Actually, it's right. I don't have it here. Testosterone is carried by this binding globulin in the blood because testosterone is a steroid and it's lipophilic or hydrophobic. And it goes down to the nurse cells and then is bound by androgen binding protein. And this androgen binding protein, coupled with the testosterone, turns on some genes that promote the formation of sperm and things like this. All right, so that's the androgen binding protein. All right, this is where we by default, right about this right here, we're indifferent. We're neither male nor female. However, we're going to go the female direction, unless told to do so by the SRY gene. Told to do what? Told to go the male direction. We all start out as females to begin with, and we would go the female direction, except for we have an SRY gene. Now what happens is downstream from the SRY gene, it's not directly activated by it, but indirectly activated by it, we make this Mullerian inhibiting factor. All right, this causes regression of these paramesonephric ducts that would become fallopian tubes. So we cause the fallopian tubes to regress. That's what we cause here. And what do we form? Well, we form a glans penis, we form a penis, we form scrotum. This is what we basically develop as a male. But we have to make this Mullerian inhibiting factor for the uterine ducts to regress. Or they're actually not called uterine ducts yet, but they're called paramesonephric ducts, which would have became uterine ducts. This is spermiogenesis. Remember, spermiogenesis is just your spermatids becoming mobile or capability of swimming. And this is a sperm that you learned in lab. Okay, this is the epididymis. The epididymis is a place where we store sperm. Later on, I'm going to tell you how much fluid is added at each step along the way. I might as well tell you right now, and then I'll mention it again later. Your nurse cells and your epididymis add about 5% of your seminal fluid. Later on, the sperm in the semen, the sperm in the semen encounter a seminal vesicle, well, actually paired, two seminal vesicles. they dump in most of the seminal fluid. They dump in about 60% of your seminal fluid. You hit them first. As the sperm are swimming up these vas deferens, the seminal vesicles are the first ones to dump more fluid into the semen, and they dump the most. They dump 60%. Right after the seminal vesicles, you encounter prostate glands. One prostate gland, that's not glands. And that dumps in, probably 20 to 30%. Let's make it 30. Let's just call it 30. It's an approximation again. And then ultimately you hit this, where are we at, 95? Yeah, we hit this bulbourethral gland. Your book probably says less than 5, but the way our math is, it's coming out to be 5. I'll say less than or equal to 5. And then the bulbourethral gland dumps in another 5% of seminal fluid. Ultimately, the seminal fluid is basic. Ultimately, this one's acidic. Be careful. Not all of them are basic. The prostate secretion is slightly acidic, but ultimately the seminal fluid is basic. It has fructose. It has a clotting enzyme. That's why semen is coagulated. It's called vesiculase, by the way. I'll spell it out for you next slide or in a couple of slides. It has a clot buster called fibrinolysin. That's why semen goes through what we call liquefaction. It has an antibiotic. It's called seminal plasmin. Seminal plasmin. That destroys E. coli. It has citrate. Oh, it has more stuff too. It has some other things from the prostate gland like PSA. You can say PSA and alkaline phosphatase and things like this. It just has a bunch of stuff in it. Ultimately, it's basic though. The fructose is the sugar source for the sperm to swim. I'll talk about vesiculase or the clotting enzyme in a second. I'll talk about fibrinolysin or the clot buster in a second. So this is what the seminal vesicles add. Initially, it's in the epididymis, and this is where we store sperm. The sperm go through what we call capacitation in the epididymis. Capacitation is the final step of maturation. You're probably going to throw your hands in the air and you're going to say, I thought spermiogenesis was maturation. Well, it kind of is. But the reality is this. Your sperm aren't ready to fertilize an egg until they go through capacitation. Capacitation starts in the epididymis. However, it doesn't even finish until the sperm hit the vaginal canal. So in other words, sperm aren't really ready to fertilize an egg until after copulation or intercourse, because the sperm finish capacitation in the vaginal canal. And that's kind of cool. You've probably never heard that before. Probably a lot of this that I'm talking about you have heard before. That may be something new to you. But capacitation starts in the epididymis and then finishes in the vagina. This is showing you that the vas deferens are lined by stereocilia. The sperm can swim, but this helps them along. Oh, actually, this is the epididymis. The epididymis is continuous with the vas deferens. The epididymis and the vas deferens have this stereocilia. This helps the sperm along, so they don't have to rely solely on their flagella beating. Stereocilia will move the semen and sperm along through the vas deferens. This is a cut open picture of it. You can see that there. All right, capacitation. So capacitation is to become motile and functional. Able to fertilize. And capacitation begins in the epididymis and finishes in the vagina. Finishes in the female reproductive tract. All right, so capacitation is not complete until after copulation. The seminal vesicles here. So remember the epididymis and the vas deferens. The nurse cells and epididymis add 5%. Your seminal vesicles add 60%. Here's your prostate, and that adds another, I think we said 30 before, 20 to 30. We'll put 30 here. 30% of your seminal fluid, of your semen. That's 95. That means bulbourethral does 5. There you go. Okay. The clotting enzyme is called vesiculase. Vesiculase causes coagulation of the semen. And then, and this is part of a fertility test, coagulation of the semen, and then it becomes liquid again. And that's called liquefaction. And that has to happen in 15 to 30 minutes to be fertile. So if you work in the lab, you'll send a male into a room to collect some semen, some ejaculate. They bring it back to you, and the lab takes it in. And one of the things they do is they time liquefaction. Liquefaction has to occur within 15 to 30 minutes. So you start your stopwatch, so it's this white, opaque, coagulated semen. You start your stopwatch, and 15 to 30 minutes later, it needs to be a watery, clear semen. That's called liquefaction. That's due to fibrinolysin. That's what that's due to. Fibrinolysin is the enzyme that does the opposite of vesiculase. Vesiculase causes coagulation. Fibrinolysis breaks down the coagulated semen. Well, it breaks down the fibrin. Vesiculase changes fibrinogen into fibrin. I should write that. Fibrinogen into fibrin. That's what vesiculase does. And the fibrin is why your semen is coagulated. And then fibrinolysin breaks down the fibrin. And that's why your semen becomes liquid again 15 to 30 minutes later. All right. This is showing you the ductus deferens, cross-section of it. There's smooth muscle here. That's quite important. All of this smooth muscle, because during ejaculation, the smooth muscle is going to contract. So ejaculatory contraction, and it moves the sperm along. Ejaculatory contraction. All right. Thick, smooth muscles around the vas deferens. This is showing your seminal gland. It's making 60% of your semen. What's this showing you? Prostate gland. The prostate gland's secretions are slightly acidic. All the rest of them are basic. And overall, your semen is slightly basic. This is your bulbourethral gland, a basic secretion. The bulbourethral gland will actually secrete some of its secretion into the urethra even before ejaculation. And this pre-ejaculatory secretion, and by the way, your seminal vesicle, all of them really will, but the bulbourethral is the important one because this pre-ejaculatory, can't spell, pre-ejaculatory secretion neutralizes the acid of the urethra. Now, why is your urethra acidic? Because you urinate out of it. And urine is a pH of 5 or so. So by the time your sperm hit your urethra, you've already neutralized the acids from any residual urine. So this pre-ejaculatory secretion from the bulbourethral is a good thing. Neutralize that, any residual acidic urine that's in there. The seminal plasmin right here, that's the antibiotic. This comes from your prostate gland. You can see it says your book tells you 20 to 30. I use 30 as an example. It's just, that's it, 20 to 30. Men usually at some point in time in their life will suffer from benign prostatic hypertrophy. That's an enlarged prostate. You can see that if this prostate enlarges, it's going to narrow your prostatic urethra and you'll have a hard time urinating. When you go in and you take out the prostate, especially if it becomes malignant, you take it out. Quite often you'll you'll hit or nick, the doctor will, the pudendal nerve, and that leads to erectile dysfunction. Nowadays, they have a robot that does the surgery, and they have, it used to be that 70% of the time you have erectile dysfunction. But now it's only 30% of the time do you have erectile dysfunction. See, they swapped those statistics. It used to be 70% of the time you ended up with erectile dysfunction, and 30% of the time the surgery went well. Now it's only 30% of the time do you have erectile dysfunction, and 70% of the time the surgery goes well. And that's because the robot's better at not hitting your pudendal nerve than the human is. All right, this is the bulbourethral gland. You can see that there. And then this is good. It's the same numbers I used. This is the same number I used, 20 to 100 million sperm per milliliter, and there's 3 to 5 milliliters per ejaculate. All right. And that's the overall. Now this is the erectile tissue, so this is how a male gets an erection. The veins that go into here are usually constricted. When you're flaccid, they're constricted. So when your penis is flaccid, those veins going into the erectile tissue are constricted. When your penis becomes erect, I'm sorry, let me say that again because I'm screwing up veins and arteries. The blood flow into your erectile tissue is slow, and the drainage of them is fast when you're flaccid. When you become erect, the blood flow into your erectile tissue speeds up, but the drainage of them slows down, so the blood stays in your erectile tissue. Your erectile tissue becomes engorged with blood, and that is what causes an erection. And this is caused by, well, let me just give you the deal right now. We always say point, shoot, score. Point, which is the erection, is caused by the parasympathetic nervous system. That's why we say point for parasympathetic. It actually uses nitric oxide as a neurotransmitter. Shoot is the ejaculation, and that's the sympathetic. And that's sympathetic. I'll abbreviate that because I don't have enough room. And score is the somatic, because part of ejaculation is also muscles of the urogenital diaphragm. All right, so that's erectile tissue. You can see that you have one corpus spongiosum. It surrounds the urethra. However, you have two corpora cavernosa, and they're on each side of the penis, and they both have an artery in the center of them, feeding them. All right, this is the penis. This is the dorsal side of the penis right here. I know it sounds funny that this is the dorsal side, but it's only because we have humans have a pendulent penis. If we didn't have a pendulent penis, it'd be easy to see that this is the dorsal side. This is the hormone control of the male reproductive system. We've already talked quite a bit about it, but let's just do it again. GnRH comes from the hypothalamus and tells the anterior lobe of the pituitary, the adenohypophysis, to make FSH and LH. Well, what do? FSH goes to the nurse cells and it says, hey, turn on spermatogenesis for us. That's what FSH does. LH goes to the interstitial cells and therefore is sometimes called interstitial cell stimulating hormone and says, make testosterone. Let's go down to the next slide. FSH is coming down to the nurse cell and it's saying, hey, turn on spermatogenesis. Meanwhile, the nurse cell makes androgen binding protein. I'm looking for it so I can circle it for you right here, which binds testosterone. Here comes the testosterone from the interstitial cells. And this testosterone also helps the nurse cell promote spermatogenesis. But meanwhile, while all of this is occurring, the nurse cell also makes inhibin. And inhibin goes back up and shuts down GnRH and such shuts down FSH. So spermatogenesis is self-limiting or self-regulated. By the way, testosterone itself negatively feeds back up to the hypothalamus and shuts down GnRH. So that's self-limiting, and there's a negative feedback loop for that. So what do these things do for us? Well, the FSH leads to spermatogenesis and spermiogenesis. See, I don't know why they say it like that. Spermiogenesis is really part of spermatogenesis. So well, I don't care how they really say it. The FSH promotes spermatogenesis and spermiogenesis, and the testosterone does sex drive, growth, secondary sex characteristics like beards and axillary hair and stuff like that. And this is still secondary sex characteristics. And that's it. Male is pretty easy. Female is actually not that bad either, but we have to talk about two cycles, the ovarian cycle and the uterine cycle. Otherwise, it's not too bad. It's real similar. Testosterone is a steroid hormone, so that must be carried by this binding globulin, this gonadal steroid binding globulin. Gonadal steroid binding globulin. Albumin also carries some of our testosterone. We men do have some estradiol. There's an aromatase enzyme that converts testosterone to estrogen. So yeah, we have a little bit of aromatase that converts testosterone to estrogen. But we don't convert a lot of it. We make very little in a day. 2 nanograms per deciliter is very little. Most of it comes from the aromatase enzyme, and we don't even have much aromatase enzyme. But the nurse cells do secrete some estrogen directly. Not much. I mean, the total estrogen is the 2 nanograms per deciliter. But some estrogen, about 1/3 of it, is secreted by nurse cells. The rest is converted by aromatase to estrogen. And that's it, folks. Next is the female reproductive system.